Okay, so we've got lots of great science we're going to show today. You don't want to hear from me, but there's just a couple of thoughts and reflections that I want to share with you. So we're going to talk a little bit about data, a little bit about dementia, and a little bit about just getting things done and not letting people say, you can't do that. So the first thing I want to say is welcome to Glasgow. Willkommen, bienvenue, welcome, or as they say around here, all right? I know how difficult it is in all of our busy schedules to take a day off, to come to a city for this kind of thing, to give yourself the luxury of a day of just enjoying science. So thank you all. Thank you all for coming. And I know why you're here. You're here because you get it. You understand how important dementia is, and you understand what data can do. I've got some slightly mundane housekeeping that I do have to get through. I'm too myopic to see the screen there, so I'm going to have to turn and look here. We're not expecting a fire, so if there is a fire alarm, we all leave and we congregate at Central Station. The toilet facilities, I actually don't know where they are, but they'll be signposted, I'm sure. When you get bored with me and you want to check your email, the Wi-Fi code is there, customer first, all in lowercase. We want to promote this event. This is a celebration, so we want to promote it. We have our wonderful comms officer from the Brain Health Arc here, Millie, and also the comms team from GAP are going to be helping celebrate what we're doing. So we'll be taking some photographs, we'll be recording the talks. If you're not comfortable with that, please do let us know. Because all of this has been done at speed, the data you're going to see today are fresh. They've not been through scientific peer review in a journal. So let's treat these data as new and, just be careful about how you share it and how you interpret it. We have crammed so much into each session, we're going to have to play it by ear to see if there's time for questions after each speaker. And we're going to leave that at the chair's discretion. But what we've done in formatting the day is give lots and lots of time for coffee, lots of time for you to engage with the people who are speaking. So don't feel that you'll be disadvantaged. If you want to speak to any of our speakers, there'll be opportunities to do that. We know a whole day like this can really be quite a lot. It can be quite overstimulating. So we are going to keep the poster room as a quiet room. And if you need to take a little bit of time to decompress, that's what the room is for, and you're very welcome to do that. And the last thing I want to say, if you're enjoying any of the stuff you see today, there's lots more to come. So please do join us. Join up for the mailing list of the Brain Health Arc. The website is there. You've got a Wi-Fi code, so you can do that right now on your phones while I'm speaking, and you'll hear about all the other brilliant stuff we do in the Brain Health Arc. And let me just do a bit of shameless plugging of the Brain Health Arc. So we're funded by the Scottish Funding Council, and our remit is very much about improving the capacity around brain health and dementia research in Scotland. And that's not just about delivery of research, it's about the whole research ecosystem. So what have we been doing in the last 18 months? We've been working with government. Research is great and it's academically interesting, but the real power comes when it starts to shape policy. And we've been doing exactly that. And actually, as an indirect result of this data challenge, I'm now chairing the Scottish Government Data Group on dementia. We've also been trying to create a community. In Scotland, we have lots of passionate, enthusiastic researchers, but there was a bit of a risk that they were working in their own geographical silos. So through meetings, through an online community, through our newsletter, we've been trying to bring everyone together, and it's meetings like this that show that I think we've been successful in that. We realise the future of dementia research, certainly not an old fossil like me, it's people that are coming in, people that are new on their research journey. So a lot of what we do in the Brain Health Arc is about supporting those people who are new to research. And we've been doing that through training, we've been doing that through mentoring, we've put our hands in our pockets and we've actually gave some money as well to help people start on their research journey. And I think one of the things you'll find with the Brain Health Arc is any resources that we get, we make available to the whole community. And testament to that is this day today. We were given a fantastic data set. We didn't squirrel it away. We made it open to all of the UK and all of UK collaborators. So that's how we roll in the Brain Health Arc. If you want something completely different, the meeting we're running next month is about the intersection of the creative arts and brain health. In the Brain Health Arc, we want to bring in new ideas, we want to bring in new energy. So this meeting in Edinburgh, we're going to have lots of artists, and we're going to have lots of people working in the brain health space. We've still got spaces, limited numbers, but there are still spaces if you want to come and join us in Edinburgh. Brain Health Art partners with the Scottish Dementia Research Consortium, which is the research arm of Alzheimer Scotland. And just while I have the floor, before you throw me off, I'm going to do another shameless plug for our annual meeting, which is in Edinburgh. It's a meeting that's co-designed by people living with dementia. So slightly different to some of the scientific meetings you might have been to, but really a fantastic meeting and a nice way to start your summer. Have we been successful? It's very un-British to talk about how successful you've been, so I'm going to let other people say how successful we've been. There was a themed issue of Scientific American last year, all about dementia. There was one country had a whole section. It wasn't America, it was Scotland. There was a recognition that what we're doing in Scotland is novel, and it's exciting, and it's got potential. The data challenge has also generated an awful lot of interest. So the data challenge featured in Radio Scotland. It featured across all of the red tops over the summer. It's featured in the Scottish Government. It was mentioned in Holyrood as an example of what can be done when people come together. And there was a whole feature and interview with me on That's TV. I've not heard of it either, but it's there if you want to see it. So that's the data challenge, and that's the ARC. I want to just, if you will, just forgive me, I want to just take 5 minutes to think a little bit about data, because today's all about data. And I want to think, if we don't have data, what have we got? We've got opinions. And I would say in the year 2025, one thing we don't need more of is opinion. We need fact. And we need to move from opinion to something that is based in hard fact. And if we take that data and we apply it to a problem, what we start to get is knowledge. And then if we take that knowledge and we share it and we apply it and we start to do something with it, what we start to get then is wisdom that can make a difference. And we can use that wisdom to go full circle, and then we're moving really from opinions to hypotheses that we can test. And how do we test them? We test them by generating data. And we have this virtuous cycle that I think we're going to see today. Some of what you're going to see today is very much hypothesis generating, and some of it is slightly more definitive. Here's a little hashtag you will have seen. All through the pandemic, you'll have seen social media materials saying data saves lives. And then that became the title of the UK White Paper on how healthcare data could be used. And I like this because it shows the power of data, but it also reminds us each datum is a life. It's a person. In our case, it's a person living with dementia. And we can't forget that. We can't forget the power of the data comes from those people that have contributed the data. Data does save lives, but it does other things. And it has done things that have very much been the engine for why we ran this challenge. Data builds teams. By making this data set open to everyone, we were able to bring people together. When we looked at applications to access the data, we asked, tell us about the new collaborations you're forming. And applicant teams in the room will know if you weren't forming new collaborations, we did a bit of matchmaking for you, and we created new collaborations. Data also fosters equity. It's not a level playing field in research, let's be honest. But data doesn't care. Data doesn't care what your H-index is. Data doesn't care if you're from a Russell Group university. So data can bring people together and level things out. And data drives innovation. Again, when we asked people to come and apply to use the data, one of the things we looked at was, are you bringing new ideas? And you absolutely did, and we're going to see that today. We're going to see those new ideas. But I think most importantly, data's quick. And let's just explore that a little bit. Because traditionally, research is slow. And unfortunately, dementia isn't. Dementia doesn't wait. Dementia just progresses relentlessly. So let's think of a scenario where you have some great ideas for using data, but then you hit a brick wall. You have to apply for ethics. You have to get through GDPR. You have to sign off material transfer arrangements that have to be signed by every partner. And it's a nightmare. And it takes forever. And lots of people just drop out at that point. The people that get through that then are given a data set that's often very difficult to work with. You don't really know the structures. You're maybe having to create your own data dictionaries. Again, very slow, and lots of people drop out. So the number of people that come through that to actually be at a point where they have results that can be shared is modest, and there's huge inefficiencies. What we've done in the challenge is take out all the roadblocks. We've let researchers do what researchers do best, which is research. All of these other bits, we did for you. I say we, it's actually the rest of the team, I didn't do it. But we took all of those stopgaps away so that you can get your research done and get it out there. The last thing I want to talk about is this concept of open data. Open data, we love it. Funders mandate it. We all talk about it. To paraphrase Animal Farm, some data are more open than others. And let's just think about that. So you have the data, you don't really want to share it, traditionally. So you say it's open, but it's open if you can afford it. It's open if you can pay the access fee. Or it's open if you're willing to start emailing all the contributors to get their permissions. Or it's slightly open, but some of it is a bit too interesting, so I'm going to keep that and you can just have the boring bits. Or it's open, but we've coded it in a way that it's completely impenetrable to anyone that doesn't work in our university. Or we've formatted it in a way that you need very specialist software or very specialist platforms. So there's open and there's open. What we did with the data challenge was properly open. And I think everyone in the room will agree with that. And that gives us speed. And this honestly is almost my last slide. And I just want to finish by reflecting on speed. So in the summer of 2023, we had colleagues from GAP come and join us in St Andrews, and over a very lovely meal, they said, we've got this data set. Would you like it? To which we said, affirmative. But there was caveats. The caveat was, if we were given the data set, we had to make it open. And that's what we did. And from July to a launch event in Glasgow in February, we did all the due diligence, we did all the ethics, we did all the GDPR to make it completely open. We launched the call, and now here we are in March 2025, with teams from all over the UK have taken that data and have done some fantastic science that we're going to hear about. I would challenge you to show me something else in biomedical research that has went at that pace. If I may, I think it's pretty impressive. I am now contradicting myself because I'm going over time, and this whole thing is about being quick. So let me just finish with this. Here's what we have planned for the rest of the day. We've already had a fantastic poster session, really impressive presentations from all the people that brought posters. I know the Heathrow fire has meant that some people can't be with us today, but for the people that have came, really very impressive. If that's going to be the level of quality throughout today, it's going to be good. The session that we have now are a series of talks about data. We're then going to have some quickfire talks using the BioHermes dataset. We're then going to have Professor Will Whiteley giving our keynote lecture about the work that he's doing to make NHS data open and available for research in Scotland. Lots and lots of breaks. We'll then have a session of talks, slightly longer talks using BioHermes data. We'll then hear from Matt Clement from the AD Data Initiative. We'll then hear from John Dwyer from the GAP Foundation. We'll have our final talks, and then the bit you've all been waiting for, we'll announce some prizes. And then if you're able to stay to the very end of the day, we're going to have 2 moderated discussions to think about how we might use data in the future and to think about what a data challenge may look like in the future. So that's your menu for today. I now need to consult the more detailed menu to remind me what comes next. And our next speaker, I'm very pleased that we have representatives from LifeArc here. And our next speaker is going to be Andrew Leach, who's going to tell us a little bit about the work that LifeArc are doing. Enjoy today, everyone. Thank you.
Is that better? Okay, great. I'm not infectious, but I have some throat issues. So I'm here as we just heard, with actually several colleagues from LifeArc, and I see my role here as hopefully to initiate some thoughts in your collective minds. That means you want to go out and talk to some of my colleagues here about some of the things that LifeArc is doing and ways that we might be able to both interact with this group and potentially help you translate some of the research that you're doing closer to patient impact, because at the end of the day, that's what LifeArc is all about. For those of you who've not heard of LifeArc, and I appreciate there might be quite a few of you, some of you may have heard of our previous existence as the MRCT. So we were set up to essentially translate the research outputs from the Medical Research Council closer to patient impact. We changed the name to LifeArc in 2017, and the key event for us was monetization of Keytruda, which I won't go into, but essentially that enabled us to become a pretty substantial self-financing charity, so a medical research charity. As you'll see in a minute, we do quite a lot of funding of research and translational efforts in particular disease areas, including dementias and neurodegeneration. And we've been undergoing quite a significant amount of change over the last few years with our sort of new strategy, and indeed our new CEO who joined us last year, Sam Barrell. So here's Sam, and if I was to try and sort of summarise what we're about, then it's translating science into impact. We have a particular focus on rare and ultra-rare diseases, but also those more generally with underserved conditions. So we have a global health initiative as well as our interest in rare diseases. And a little bit dark, essentially we're looking to help move ideas and research closer to patient impact, whether it's in a sort of earlier phase, which is where traditionally we operated, but also we have ambitions now to get more involved in the sort of later preclinical and clinical phases. And there'll be more coming out about our particular activities and efforts in that area. So we're a translational organisation. We have a lot of expertise. Really interested to hear what Terry said about open data. I came to LifeArc from the European Bioinformatics Institute, which is essentially the home of big data for biology and the life sciences. So I'm absolutely on message with that concept of open data and the value and power that can bring. In terms of what we can do, then we have a science organisation. We have about 120 people. We have labs in Stevenage. We have offices in London and also in Edinburgh. We get involved in providing ultimate advice, and we do provide quite significant amounts of funding, again, in particularly directed areas. We're looking to complement what is going on already, so we're not looking to compete overtly with other funders, be they government, be they investment from the commercial sector or other charities. But we work with a lot of organisations of those type to maximise the outputs and the impact of what we are able to fund. And just one other sort of piece of, as it were, the sort of corporate level, we have quite ambitious plans to consolidate our Stevenage and London sites into one location in the Knowledge Quarter in London, in a new building that will take a couple of years to actually come to fruition. But we really see that as a sort of focal point for translation in that sort of rapidly growing area of the country. So I am the head of data sciences at LifeArc. Again, as I mentioned, we have several of my team members here, together with Giovanna, who is our resident expert in neurodegeneration and other similar conditions. And what we are doing is working with both our internal colleagues, lab-based colleagues, as well as with many other collaborators, and as I'll say in a minute, in particular in sort of the use of healthcare data, in bringing our expertise to help do the kind of research that everybody else is doing here, but also with a particular view to how we translate that into patient impact. We cover the range from sort of chemistry, computational chemistry, bioinformatics, but also in particular around the use of clinical and healthcare data. And also, for example, my colleague here, Sam, over in the corner, heads up our AI ML function. I'm sure there's a few who might be particularly interested in that, want to have a chat with him about that in detail. So the sorts of things we do, for example, in the molecular design area, we have a particular focus in targeted protein degradation. We have chemists in the lab. Again, I can explain more about this if you're interested in talking to me afterwards, but this is quite an active and very interesting area. in terms of drug discovery and chemical biology. We have a long-established antibody platform at LifeArc with our lab-based colleagues, and we are working closely with them to bring our expertise to really help sort of enhance what they're already very capably doing, both in terms of antibody humanization as well as antibody discovery. And just as an example, again, there's more information on the web, but we have a antibody discovery platform called BSmart, which allows us to generate humanized antibodies from a mouse model. There's a sequence analysis and selection part of that process, which we're working with our colleagues to identify the best candidates from that large repertoire of initial antibodies to take forward to the eventual human candidate. And then, perhaps most relevant to this group, we have made and are making quite significant investments in what we call clinical data, that's with healthcare data. We are a key partner with Our Future Health, which again might be something that people here have heard about, and something that we're really keen to do is to facilitate conversations between groups such as this and organisations like Our Future Health. And I think that's one of the key things that LifeArc is able to do as a charity. We don't come with some of the perhaps baggage as it might be of some maybe more sort of commercial organisations. So there are maybe doors that we can help to open that might be perhaps more challenging for others. But as part of that, we've been growing the team in this healthcare data analysis area. And just to give you one example of, again, this isn't a dementia-related disease, but this is a rare disease that just illustrates, I think, quite a lot of the kinds of analysis that I'm sure we'll be hearing more about during the meeting today. So this is a rare pain facial condition. We're working in collaboration with one of the country's leading experts in this, and this is very much the kind of operating model that we have. We're bringing our data science expertise, capabilities, resources, and sometimes budget to help, let's say, a clinician who has access to patients, to data, but not quite sure what to do with it or how to get the best out of it. And in this case, the data is coming from CPRD, we're on the national level data resources, but I'm looking to answer very specific questions about, for example, the geographical prevalence of this disease. That can then be used, for example, to decide where to maybe set up regional centres that will get the maximum sort of value for the patients with that particular condition. So coming then on to obviously the topic of today's talk, this is a slide that Giovanna has kindly provided to me. It states many of the things that I'm sure we're all very familiar with. And just again to reiterate, LifeArc's interest and investment in this area. 2 weeks ago we had a one-day summit in London, as you can see, translational summit on MND and rare dementias. That really is the sort of focal point that we've chosen to sort of aim for when it comes to, again, that sort of unique contribution that LifeArc can make. But clearly that's in the broader context of the neurodegenerative and dementia diseases more generally. You can see here we've got 3 patients with MND as part of the day-long event, and really sort of exciting and I think invigorating sort of day for all concerned. These are just some of the key takeaways that I borrowed from Paul Wright, who is our translational challenger as a sort of lead in this area. So these relate to research questions around heterogeneity, practical questions about reducing time to diagnosis, the whole question of clinical trials, which, as I mentioned earlier, is something that we are going to be more actively involved in. But clearly there's a sort of patient voice, a patient sort of communication, engagement piece, as well as you see here, collaboration, data sharing. So again, I think that's going to be a common theme. And I just want to finish up then with our this is the most data slide that I have. Thanks to Elenia from the team who is here today. We're working with Valentina and her team to do some proteomics analysis on the BioHermes001 data. Really interested to see there's at least one poster on this as well, so I'm sure there'll be conversations that we'll be having as a follow-up. I'm not going to go into the details of this. If you want to find out more, then ask Elena or others about that. But just to say, we're also involved in BioHermes001, which I'm sure many of us in this group either are or are aware of, and we're looking forward to the output both from the BioHermes001 as well as the successor project. So with that, I'll finish up with acknowledgements. With colleagues at LifeArc, together with team and Jana, who we've been working with. And with that, I'll stop. Thank you.
So our next speaker is online. The whole idea of sharing data, I'd love to say, was my idea, but it's been around for a while. And another very good example of repurposing data and sharing data and using data for new discovery is the Dementia Platforms UK data portal. So I'm very pleased that we have Simon Young as a remote speaker who's going to tell us a little bit about DP UK data. Simon, are you with us?
I am with you, Terry. Can you hear me all right?
Loud and clear. Over to you.
Okay, fine. First of all, thank you very much for the invitation to talk. Thank you to Terry and the Brain Health Arc, and very sorry that I'm not able to be in Glasgow with you all today. There's lots of people that we work with very closely in the room, and we came to present at the February 24 meeting, so it's a shame I wasn't able to come. I'm going to talk about the data portal, as Terry has already stated, and yeah, just be clear, I'm Senior Programme Manager for Dementia's Platform UK. The data portal has been in operation now for just over 8 years. DP UK has been running for almost 11, and really pleased how the data portal has grown and continues to grow. Each year we have more and more applications to use the data portal. Have currently 85 different data sets sitting there and over 1,200 people using it from around the world. And to Terry's points, what he said before, we very much want to try and speed up access to data, to make it open and available to researchers, and we do not charge. It is free to use. We, however, operate in a safe environment as well. So we've got to balance that openness with safety. And we adopt the 5 safes principle, and primarily making sure that we have safe data. It is anonymized, it's not identifiable. It's in a safe setting so that it can't be accessed inappropriately. We have safe outputs. So we check what information is going out. We do not let people take raw data out of the data portal. People analyze the data on a virtual desktop, which is secure, and it's safe people. The people that are accessing it are checked to make sure that they are legitimate researchers. But I hasten to add, it's not just academic, it is being used by industry people as well, growingly. We have a variety of discovery tools on our website, ranging from top-level information down to the level of information, the level of variables that are there, and interactive tools to enable researchers to determine whether data sets hold the kind of information that they are looking to use. Behind that, we have an ontology, a structure to the data. And we determined that there wasn't anything available, so we've decided to develop something ourselves and use that to enable more informed data discovery and also ability to combine data sets more easily. So the ontology that we've developed has...
Simon, I'm really sorry to interrupt. Yeah. We're still on your first slide here in the room.
Okay.
So if you click on slideshow again, it's the icon beside the slider. That should work. And then you might need to just change the screen that you're sharing from.
Any better?
No, why don't we keep this view and
or
scroll on this?
If you just scroll down on your presenter view, then we'll keep up.
Do you want me to start re-sharing, Terry? Is that best?
I think if you use the scroll down function at the very far side and just go through your slides that way, then that's what we're seeing in the room.
So I'm not sure I know what you mean by the scroll down function, unfortunately.
We can keep it like this. We see your slide right now, so it's fine.
Yeah.
Which slide are that? Do you have two screens?
So if you have two screens, Simon,
Yeah.
if you change the screen that you're sharing, that will work.
Okay.
Up the top, there's probably an item that says display.
Yeah, I've changed that twice. Is that any better?
Let's just run with it.
Yeah.
So
which slide can you see at the moment then, Terry?
Creating research-ready data for human cohort.
Okay, right. That's the right one. I will. Hopefully, you'll continue up, but let me know if it's a problem.
Yeah.
So, okay, the ontology then that we've developed has got 4 levels. At the top level, there's 18 themes, and then we drill down to domain, where there's 200 or so, and then family, where there's about 700-800. And then objects are the actual data items themselves in the thousands. So that helps, as I said before, data discovery, data understanding, but ability to work across multiple data sets in a more informed way. So that's standardization of the data that is available. But we've also worked on harmonization as well. We've developed a data set of 40 variables that we're looking to harmonize across the data that we hold on the DPOK data portal, but also across other data sets as well. And we're working very closely with the AD Data Initiative, the data that they're holding on their AD workbench, to provide this harmonization and the standardization that I just mentioned before, and also working with other platforms on that. That harmonization is then used in the interactive tools, and this is Cohort Explorer that is available on our website. Anybody can have a look at the information that sits there to determine whether the information held within the data sets are appropriate to the kind of research that you're looking to perform. In order to make sure that researchers have the necessary skills to be able to analyze the data, we've been running for a number of years now various training workshops, primarily 2 key areas, one being academies, which we hold twice a year, and we've got one coming up in the next few weeks. There's time to apply to join that if anybody in the room wishes to. It's focused more at the elementary level, so building basic skills to analyze the data. We have a more advanced one later on in the year. We very much want these to be available to as many people as possible, so we keep the costs as cheap as possible. We do have to make some charge because of the people that are presenting require some degree of payment, but we keep those costs, and compared to other commercially available training courses, they are very cheap. The other training events that we hold are datathons. Very pleased we worked with Terry to have one a year, maybe 2 years ago, and these are free. These are for anybody to attend, and groups of researchers are put together and they analyze data, actually real data on the data portal, and to answer a specific question or a topic, and then swap notes at the end of a week of working on those data, and some go on to analyze those data further and write up papers on what they've found from those data. In addition to phenotypic data on the data portal, there's a lot of images. We've over 135,000 brain images from 20 different data sets. So raw images available if you have the skill set to utilize those, but we have different processing pipelines in place that generate image-derived phenotypes from those data. So if you're interested in what's in the images but aren't particularly interested in doing the analysis yourselves, there is the ability to get hold of the information from those images. And similarly for genomics, we've got a range of different genomic information from ApoE status all the way through to GWAS and others. And again, putting in place data processing pipelines to analyze the data and polygenic risk scores that I'm sure we'll be hearing more about later on in the day. What we're trying to do then within the data portal is give this virtual laboratory. We've got a lot of survey data, we've got a lot of image data, we've got a lot of genotypic information, and growing amounts of omics data and a growing amount of digital data as well. One area that we've been working on for quite a while, and pleased to say it is getting close to fruition, is bringing in electronic health records, and I'll mention more on that in a little while. And also within the data portal, we do have additional parts of the DPUK program being shown as well. The experimental medicine work that Terry and others are working on, results from that are increasingly being shared within the data portal, and the registers that we've got available for people to recruit from are available within the data portal. We also have a growing area of specialist hubs, which are essentially special areas that are focused in a particular area of research. And I've given a few examples on the screen there, which I hope you can see. So from traumatic brain injury to motor neuron disease to stem cells, and essentially these are dedicated areas that have their own look and feel, their own discovery tools, but they can build on what DPUK has already developed, and it is a quick way in for them, but it grows the family of different capabilities within the data portal. We also have various collaborative spaces available. Now, these are essentially for when people haven't quite got to the stage of being able to share the information. Perhaps it's still being collected, perhaps it's still being worked upon. But making dedicated areas for the team to analyze those data across institutions, across countries potentially, and share that analysis with a view at some stage, probably to be making those data available to wider researchers. One example of that is a meeting I was at yesterday around CSF testing in memory clinics, and how that can be brought together, trialing it across 4 NHS sites in the first instance. So sharing those data on the data portal in a collaborative space. Once the learning has been gained from that, then we can feed that back to the NHS to hopefully have a model for how that can be rolled out more widely, but also share the data that has been developed can be shared to other researchers. And then just thinking about where we're going. I've mentioned federation briefly earlier, but we have federation in place with the AD Data Initiative and the data sets on the AD workbench. But we're rolling that out with additional technology that's been developed within Swansea University to other endpoints as well. So DP Australia and DP Korea have their own repositories, and we are looking to share that information so that researchers can just come into one entry point, one front door, and then have access to data that is around different repositories. and the number of different repositories will grow. AD workbenches there, as I mentioned already. But also trying to bring in the NHS data, so the electronic health record information, alongside the research data that currently exists. So bringing molecular, cellular information along with the electronic health record information into a trusted research environment to be able to share that with researchers, so that more and more research can be performed in exactly the way that Terry was referring to earlier. And that is what DPUK is trying to achieve. Terry, that's the end of my presentation. I hope you saw all of the slides, and happy to take questions if time permits. So thank you very much, Simon. And I would just echo what Simon says. The DPUK data portal is a fantastic resource. The hackathons, especially, are a lot of fun if you want to devote a week of your life to just really immerse yourself in data. Simon, we did have a few gremlins with the presentation. Would you be happy to share your slides and we can then share them with all the delegates? I've already shared them with Sheila, so you should have them already, Terry. But yeah, very happy for you to share them. Yes.
I'm going to press on and not take questions, but there are lots of ways to get in touch with the data portal team through the DPUK website. So let's give Simon a note of appreciation. Thanks, Simon. So we're going to move on to quickfire talks now. A problem, but a nice problem, was we had so many people working with the data, we struggled to think how we're going to fit everything into one day. So we've had some posters, and now we're going to have some really very snappy 5 minutes, guys, talks for projects that are perhaps a little bit earlier in their gestation.
So, hello everyone. I'm happy to attend this very interesting event related to BioHermes Data Challenge. So I will briefly present some of our results in the project that we conducted in collaboration with the University of Manchester, King's College London, University of Glasgow, Brighton and Sussex Medical School, and Vaslui County Hospital in Romania. I'm Daniel Stamate. I work in London at University of London Goldsmiths College, and I'm the lead of the project. So basically, in our research, we could tackle two main directions, and the first of which is proteomics, neuropsychological and demographics multimodal machine learning approach to Alzheimer's disease prediction on the BioHermes study cohort. And the second direction, on which I present some results today very briefly, concerns classifying cognitive states of Alzheimer's disease with machine learning using digital biomarkers from the BioHermes study cohort. So in the first research direction we tackled, basically we conducted a study applying machine learning, developing some models on the BioHermes study cohort, in a multimodal approach to classify individuals into cognitive normal, mild cognitive impairment, and Alzheimer's disease cohorts using blood-derived proteomics, neuropsychological test results, and demographic data. As methodology, we developed models concerning from more explainable and simpler model like the elastic net to decision trees, random forest, and extreme gradient boosting as the kind of top performing algorithms. We developed optimized models to make these predictions in a five-fold cross-validation fashion. And basically, we wanted to see how stable these models are, conducting a Monte Carlo validation based on 100 iterations. As results, we evaluated these models and we saw that the top performing model is XGBoost. We obtained quite a good stability in the Monte Carlo simulation that we conducted. So we can see that models are pretty stable. In terms of looking to what kind of predictors would best influence the prediction. So we've seen, of course, predictors like mean mental state examination. And from the proteomics, blood proteomics data, p-tau was in the top 5 predictors. As a discussion, these findings demonstrate that multimodal machine learning approaches can effectively classify Alzheimer dementia cohorts. While the top performing model like XGBoost achieved the highest predictive accuracy, simpler models like ElasticNet offer a good performance and superior interpretability. And in the future, we plan to continue this kind of research, validating the models on a larger cohort and on more diverse populations, and explore explainable AI techniques to enhance clinical applicability. With respect to the second direction of research we conducted, this concerned the use of digital biomarkers because they are cheap to obtain and non-invasive compared with the more conventional biomarkers for AD. And we explored models like logistic regression, ElasticNet, random forest, gradient boosting, and XGBoost again, in a fashion of splitting the data into 80 and 20%, and doing the relevant transformations on the data to ensure maximum performance. So in particular, we've seen that in our methodology, XGBoost and in general gradient boosting methods were the best in terms of performance, like AUC, area under the curve, which is a generic measure to assess the performance of such models. And in particular, we were looking also to see in the top 20% of risk calculated by these models, how many of these patients actually were with AD. So the table number 3 here shows this kind of estimates, in particular 89.7% in the top 20 risk values were with AD. And of course, since XGBoost was the best model we had, we were looking a little bit in more detail to see at different probability thresholds what was the level of capturing AD patients. As these results demonstrate that digital biomarkers can serve along the proposed machine learning prediction modeling in this study as good indicators of Alzheimer risk and could be useful in screening processes, helping to identify individuals who may benefit from further clinical assessment. And definitely we focused on this direction because, as mentioned, digital biomarkers have a good potential in Alzheimer's disease prediction, as they are cheap to produce and non-invasive compared with the more conventional AD markers. Thank you for your attention, and if there are any questions, I will be very happy to answer them.
Thank you. And if we could move on to our second remote speaker, and that's Connor. Connor, are you with us?
Yeah, I'm with you. Can you hear me?
I can indeed. And could you share your slides, please?
Yeah, no problem at all. Can you see that? Can you see this yet?
Not yet.
Not yet. Let's try again. Entire screen. Okay, there we go. There we go. You can see that now?
Yep, we got that. We're seeing the presenter view. And can I just remind everyone online, if they're not speaking, to mute themselves, please? Okay, take it away, Connor.
Fantastic. Hello. Today we're presenting our research project, Profiling Cognitive Resilience and Resistance to Alzheimer's Dementia. And this work was undertaken in equal contribution from myself and my fellow co-first authors, Austin Dibble, who will also be talking today, and Calliope Mavramati. So when diagnosing Alzheimer's disease, we typically think of 3 clinical groups. The first is healthy controls, who present with good cognition, no Alzheimer's disease pathology, and little to no risk of developing Alzheimer's in the future. On the other end of the spectrum, we have Alzheimer's disease patients who present with a cognitive deficit and high levels of Alzheimer's disease pathology, and obviously a high level of risk of developing Alzheimer's disease. We then have this third intermediary groups, which is mild cognitive impairment, who present with a cognitive deficit. They may present with some AD pathology on the brain, and they may or may not go on to develop Alzheimer's disease in the future. Now, emerging research has suggested that there are potentially two other clinical groups that are worth investigating. The first is the resilient group. Now, these are patients who present with a good level of cognition, despite having a high level of AD pathology on their brain, and they may or may not go on to develop Alzheimer's in the future. The second group is a resistant group, and these are patients who present with good cognition and have no AD pathology, despite being highly at risk of developing groups defined. We can move on to our research aims, whereby we wanted to do an exploratory analysis to identify potentially resilient and or resistant patients within our BioHermes data cohort. Specifically, we want to take our cohort of about 1,000 patients, split them up into the three sort of classical groups, and then see if there were any patients who matched our definition of resistant or resilient. Once we had these groups defined and our patients within each group, we then wanted to explore the profiles of each of these different groups and compare between them. I'm now going to hand over to Austin, who's going to show you how we met these research aims with our current methodology.
Yep, okay. Also wondering if I should have a microphone.
Yeah, you can use one, of course. There you go.
All right. Hello, can everyone hear me?
Yes.
All right. See if we can identify them in our population of the BioHermes cohort. And okay, there it is. So one thing that we need to do is identify who has a high or a low threshold of pathology measured across all of our biomarkers. So this presents a challenge to us because there are a lot of different biomarkers in this data set. We have 8 amyloid beta measures, 6 tau measures, 8 genetic markers, and also 7 others like NFL and GFAP, which we can explore. So that's 29 in total. And so basically we have to ask ourselves, what would a high or a low threshold look like across all of those different biomarkers? So there's no universal definition that has been developed for such a challenge. And so basically the idea that we had was to let the data decide for us using basic algorithmic methods. So for those who maybe aren't familiar with t-SNE or principal component analysis, basically what we've done is we've taken all of our groups of biomarkers and we put it through the PCA or t-SNE algorithm, project it down onto a 2-dimensional axis for visualization and also ease of understanding. And oops, slowly coming. There we go. Okay. So basically, again, what it looks like is for every participant in the data set, you have 1 point, 1 circle on this chart, and then it's colored by a biomarker. For example, in this case, it's the Z-score of amyloid beta 40. And then moving on. Now, if we take all of the biomarkers that we have, all 29, and we map them together using t-SNE, and then we can basically use the k-means algorithm to define a threshold, and this is a cross-pathology threshold, so we're clustering into high and low pathology groups across all biomarkers. And so basically here in the chart on the right, you have high pathology group participants on the right in yellow and low pathology group in purple. Okay, so yes, we've used clustering to identify this cross-pathology threshold. So it's a threshold for every pathological marker that we have. For the sake of explainability and to kind of understand what the most important components of that clustering are, we also have this tree model that I'm going to show here. And this actually explains 90% of the separation in our cohort. And basically, we can see that this distinction between high and low pathological levels boils down to amyloid beta and p-tau 181, which might be well expected, but it's good to confirm. So as an example, we have the amyloid beta 40 here, and we break this into two distributions. So we have our low pathology distribution on the left and the high pathology distribution on the right. And we can see that there's the threshold line going across, which basically separates our two groups for this variable. So now we needed to do another thing in order to identify our resistant population, which is identify our low risk and high risk individuals. So this presents to us a similar challenge as with pathology, although to a lesser extent, because we have 6 distinct measures of risk, which we've derived from the relevant literature. So they're listed there. And again, we've projected this onto a 2-dimensional plane where every participant is 1 point, and this is colored by cogdad risk. And then we take all of those variables together, and we run k-means across them again, and then we get our participants who are high and low risk for dementia, at least as far as we understand. So we have a high on the right in yellow and low on the left in purple. So to bring it together, we have our resilience group that we've identified, and that's a high pathology, but also filtered as a healthy diagnosis population. And that's 108 of our 1,001 participants. And then our resistant group, which we've identified using low pathological risk, or low pathology, high risk, and a healthy diagnosis, and that is 64 of our 1,001 participants. And then again, we can basically create a basic tree of this idea of resilience, and primarily it's separated by a healthy diagnosis, which we would expect. If they're not healthy, then they have AD and MCI and are excluded. But besides that, we find that amyloid beta and NFL are 2 primary biological measures that distinguish whether they have a pathological level of pathology.
Fantastic. Thank you, Austin. Just before I go through the main results now from that methodology, here's a little table just to reference back to with some of the key identifying factors of each group. So the first thing to do is look at the breakdown of sample sizes between each of our clinical groups. And when we look into the demographics a bit further, we can see there is also a nice split between sex of male and female, with perhaps an exception for the healthy control group. But what's really interesting is when we look at age across each of these different groups. So here we can see the violin plots for the healthy control, the mild cognitive impairment, the Alzheimer's disease, resilient and resistant groups. And what's really interesting is when we look at the resilient group, we can see there's a slight skew towards an older section of the demographic. And actually it's quite similar in age to the Alzheimer's disease group. And what this could suggest is that these resilient participants, despite years of developing a high burden of Alzheimer's disease pathology, are still resisting the impairment to cognition, unlike their AD counterparts. And equally, in the resistant group, we can see there is another shift towards this sort of older age participant. And again, this could suggest that despite being at risk for several years, they are still managing to be resistant against developing Alzheimer's disease pathology and also resistant against a cognitive impairment. Now we can break these results down even further into the profiles, as we suggested before. And the first thing to look at is cognition. So what we do here is we take the healthy control as our baseline and compare each group and how far they deviate from the healthy control in their mean PCA scores on their cognition. And as expected, we can see that Alzheimer's disease is the worst performing cognition, shortly followed by mild cognitive impairment. But what's interesting, and I'd like you to focus on, is the resistant and resilient group. Yes, there is a bit of a dip, but it's probably not enough to classify them in the mild cognitive impairment, or definitely not in the Alzheimer's disease group. So what's really interesting is when we consider Alzheimer's disease pathology, such as amyloid beta. In our resistant group, we can see it's pretty much the same level as the healthy control, which bodes well for our definition of resistance, where we would expect little to no AD pathology. But then when we look at our resilient group, we see there's an increase in amyloid beta, the highest amongst all of the groups. And at the same time, we have a good level of cognition in spite of this. What's really interesting is then when we look at tau as another marker of Alzheimer's disease pathology. Again, in our resistant group, we can see a similar level to the healthy controls. And then again, in our resilient group, we can see there's an increase in tau pathology, which is pretty similar to what we'd expect in the Alzheimer's disease group, with mild cognitive impairment in the middle. And finally, we also looked at some other evidence biomarkers, such as GFAP, NFL, and the presence of an APOE genetic mutation. And we can see the trend is very similar to the tau line, where in our resistant group, we see, again, similar levels to the health control, and elevation in the resilient group that is sort of synonymous with our AD groups, and again, the MC in the middle. And so the main takeaways that we suggest for this is that we have identified these participants who, in this particular group, have a similar level of pathology and cognition as our healthy group, yet they are high risk in terms of the risk scores. And then we have this other group of participants who have a high level of AD pathology, similar to that of the Alzheimer's disease group, yet they maintain a good level of cognition. So with these results, we hope we've ticked off the first research aim in potentially identifying resilient and resistant participants in our cohort. And we're continually going to generate more and more detailed and complex profiles of each of these groups. And then in the future, we hope to identify perhaps modifiable factors, such as lifestyle or other biologically linked variables, that we may be able to change and influence and change the membership between participants of different groups. I'd like to thank you all for listening. Thanks to my first co-authors, Callie and Austin, and our collaborators, and of course, all of our sponsors and funders. Thank you.
Okay, thank you very much. Lots of people taking photographs of your slides. So I take that as endorsement by the room.
Okay, good morning, everyone. I'm Farah, and I will fire you today through the work that we've been doing at the University of the West of Scotland with our multidisciplinary team of full-time academics. And I just wanted to let you know that this is the first time our team has worked with Alzheimer's disease. We come from various backgrounds, molecular biology, psychology, and health, and really enjoyed looking at this data challenge. And our work is more hypothesis generating, as you're going to see in a bit. So our main aims and objectives were to try to figure out possibly new biomarkers that let you go from here to here, so that lead to dementia or more progressed Alzheimer's disease, and try to link these biomarkers to some demographic features. And as you all know, we had these three cohorts in the study, and so we focused on comparing different data that we have for these cohorts. And so the demographic ones, we looked at age and education. The biological ones, these are the biomarkers that most of them have already been characterized, and we wanted to confirm that they're there with this, with the participants of the BioHermes data. We also wanted to identify novel biomarkers through proteomic data that we have, and we wanted to correlate with some physical or behavioral features, specifically handedness, as you're going to see in a bit. So again, initial characterization, and some of them were already mentioned in the talks. We had that the cohort significantly differed by age, with the age going up to a mean of 74 years old for the third cohort, which is the mild AD. And even the years of education, for example, the first cohort, which was normal, had significantly the highest years of education of 15 years as compared to the others. And when we looked at these biomarkers, and I said lots of them have already been characterized, it was good to confirm that the cohorts that we were looking at had the similar pattern that has been observed before. So for example, you can see here from the MEC data that GFAP and FL were significantly higher when you go with cohorts, and the ratio of amyloid beta 42 to 40 was significantly less in the third cohort. And again, same for APOE, GFAP, the phosphorylated tau levels, again, were going higher as you go by cohort. Some of them would seem to be more significantly different earlier on, like APOE here, as compared to the amyloid beta ratios, which were more significant later on. Again, this is the p-tau data. And I told you we wanted to look at novel proteomic data, and we mined some of the proteomics data from mTheraPro. They analyzed about 800 to 900 peptides, and we wanted to get lots and lots and lots of data. However, we managed to find differentially expressed proteins that have mild changes but significant. And therefore, we're not going to show you pathways of these proteins. I have them here in the tables in case anyone wants to look at them in details. But what we found interesting in this differential expression analysis was with the complement factor H related protein 2 was the one recurring in every analysis between cohort 1 to 2, 2 to 3, and 1 to 3. And this is related to innate immunity. A little is known about the complementary system and its relation with AD development, and therefore this is why we think it's worth a further analysis. This is one of the first possible hypothesis generating data that we got. And then we wanted to try to link to some behavioral changes, and it is the handedness. And to look at the left and right-handedness, the percentage of left-handers is usually around 10% of the population, and this was kind of maintained, although there were a few differences that we did not find significant. But when we looked at the biomarkers and the difference between left-handers and right-handers for each biomarker, for example, for the phosphorylated tau 217, specifically in cohort 3, the left-handers had a much higher level than the right-handers, a significantly higher level than the right-handers. Same goes for NFL and GFAP. In addition, we looked at the amyloid beta 42/40 ratio, and again, we found significant differences in their levels between right-handers in cohort 1, but also in left-handers, both in cohort 1 to 2 and in cohort 1 to 3. So we think, and this takes me back to the idea of heterogeneity that was spoken about at the start. So it is important to consider heterogeneity in the population when it is being diagnosed. And we hope that further work with the complement system, for example, considering handedness, will help with more custom diagnostics, thinking of treatment targets, but also and having preventative measures. And we'd like to thank everyone. We'd like to thank the Data Challenge for it being available. We'd like to thank the support that was offered to us, especially from Callie and Victor from Glasgow and St. Andrews. And we hope that more research keeps getting done and more fundraising. And this is me in the memory walk in September last year, hoping to fundraise for dementia research.
Fantastic. Thank you, Farah. We're going to finish with the home team. We've got 2 talks from Glasgow. Faria is first.
Hello everyone, my name's Faria and I'm a clinical lecturer and doctor working in geriatric medicine here in Glasgow, and alongside Stelios, Callie, overseen by Terry and some other international collaborators, we've been working on a project looking at how frailty might moderate the relationship between blood-based biomarkers and clinical dementia. So frailty refers to a reduced ability to cope with an external stressor, and that's due to multiple changes in different body systems. There's different ways of measuring frailty, but one is to use a frailty index, which is a cumulative index of variables, which gives you a numerical value, which indicates how frail someone might be. With access to this dataset, we had information about the blood-based biomarkers for over 1,000 participants, as well as clinical assessments, cognitive information, functional assessments, and we used all of this to retrospectively derive a frailty index. When we're thinking about what variables we want to use, these would be general principles, so those associated with health deficits, not just with normal ageing. Generally, prevalence increases with age, doesn't saturate too early, and covers a variety of different systems, not just cognition, not just comorbidities. These are the steps that we followed, and we started off with about 40, just over 40 variables. Most of our exclusions happened at step 4, so because the variables just weren't present in more than 1% of our participants. And the final frailty index had 22 variables. And these covered a variety of symptoms, hearing impairment, visual impairment, different signs and scoring systems, looking at low mood, looking at lab markers, as well as physical measurements. We had functional assessments included as well, and comorbidities as listed there. For each participant, so each variable is coded between 0 to 1. For each participant, you add up the number, divide by 22, and that gives you a numerical value and a frailty status for each participant. Within the cohort, we had results for cohorts Group 1, Group 2, Group 3, as others have talked about, and we use this information to do some of our initial results. So, looking at the prevalence of mild cognitive impairment and dementia within our different categories of frailty, we found that mild cognitive impairment seemed to be most prevalent in those who could be categorised as mildly frail. whereas dementia seemed to be just about equally present in those with mild or moderate frailty. We then went on to a moderation analysis, so looking at how frailty status might moderate the relationship between your blood-based biomarkers and dementia. And these are just some preliminary results. So first of all, looking at frailty, it seemed to be a strong predictor of having a higher chance of a dementia diagnosis. Looking at the biomarkers themselves, when they were considered alone, there didn't seem to be a significant effect on dementia diagnosis, and we didn't find strong evidence that frailty modified that relationship between the biomarkers and dementia diagnosis either. With more work to come.
So you're being teased there. There is more to come. But the last of our quickfire talks is going to be Angelina.
Okay. Good to see so many of you, first of all. Welcome to the event. I'm really looking forward to the day. My name is Angelina. I also work with Terry. He's my primary supervisor. And I'm now a final year PhD student on the Precision Medicine Doctoral Training Program. Today I'm going to talk to you about our project where we investigated cardiovascular risk as a moderator of the relationship between blood plasma dementia biomarkers and cognitive impairment. And this is really going to be a fire talk, so if anybody has some more questions, you can always come speak to me after the session. I know we have just 5 minutes, but I'm actually going to start with a quick icebreaker. So I would like you to please turn to the colleague on your left, and then the colleague on your right, say hello, maybe give them a smile. Come on, let's get some motion in. It's good for the brain. Good. All right. Well, as surprising as this may seem, 1 or 2 of you will go on to develop dementia eventually. So it is needless to say that dementia is a mounting health challenge, and we all know this in this space. Dementia currently affects over 50 million people worldwide, and its prevalence is also expected to increase with increased longevity. We know that Alzheimer's disease is the leading cause of dementia, which has a substantial social and economic burden, not in this country, but also all around the globe. However, even though we know quite a lot about dementia, there are a lot of outstanding challenges in the field. And one of them is actually differential diagnosis, which is currently very difficult on clinical grounds alone, even for dementia experts. And this has to do with the fact that a lot of people who will have AD pathology in their brains are actually not going to manifest clinical symptoms, especially early on in the disease. And this is really where Alzheimer's disease biomarkers play a fundamental role. Now, in the last couple of decades, positron emission tomography-based and cerebrospinal fluid-based biomarkers have really drastically improved detection of Alzheimer's disease pathology. However, a very big problem, again, not just in this country, but especially in low to middle income countries, is that these biomarkers are very inaccessible, they're very expensive, and they're also perceived as invasive. So people actually are very unlikely to want to go through these procedures. And this really has led to growing attention to plasma biomarkers. We all know the hype right now. We know that these plasma dementia biomarkers offer the potential to revolutionize dementia diagnosis and prognosis. Can we leave plasma biomarkers on the side just for a moment, though? Something very important for dementia is also cardiovascular disease risk, because we know that CVD risk actually increases risk of late onset Alzheimer's disease. And we know that this increase typically happens in a dose-dependent manner. So that means that the higher the number of risk factors that a person has, the greater the probability of developing dementia. And these are things that we know about. These are things like smoking, hypertension, diabetes, etc. So this is precisely the research question that we wanted to answer with this project, namely, how does cardiovascular risk moderate the relationship between blood plasma dementia biomarkers and cognitive impairment? And this has not been done before in such a data set. So to answer this question, we developed a model where we modeled the independent variables of AB42/40, p-tau181, p-tau217, and circulating levels of ApoE4. Our dependent variable was cognitive impairment status, where we had a group of cognitively normal individuals and persons with cognitive impairment as well. And then we tested moderation by cardiovascular disease risk, which we quantified using one of the widely available tools, namely the atherosclerotic CVD risk score calculator, which includes all these risk factors that you can see here in this diagram. For our statistical model, we first, in main effects models, modeled all biomarkers of interest and CVD risk as predictors of cognitive impairment, and we then tested moderation by cardiovascular disease risk using a moderated multiple regression framework. So we used a subsample of 745 people from the Bio-Hermes cohort that we're all familiar with. We used the biomarker data from Roche Diagnostics and Lilly, so thanks Roche and Lilly for providing us with the data. And then we also included cardiovascular and demographic information, information on hypertension medication, which we needed for the calculation of our CVD risk, and of course information on cognitive impairment status. And because that's a far talk, I'm going to keep results quite narrative, but again, feel free to speak to me after this. Our main results were that blood plasma dementia biomarkers and cardiovascular disease risk independently significantly associated with cognitive impairment. However, CVD risk did not moderate the biomarker cognitive impairment relationship. So what does that tell us? Our findings suggest that blood plasma, biomarkers, and CVD risk exert independent effects on cognitive impairment, which means that we should really prioritize assessing CVD risk as a complementary assessment to other biomarker assessments as well. So just in terms of my key takeaways, we provide evidence that CVD risk and blood plasma dementia biomarkers likely exert additive effects on cognitive impairment and not synergistic ones, which essentially means that we should underscore targeting CVD risk factors as a preventative strategy for dementia. And this is good news because you just said hello to your colleagues around the table. There's 14 risk factors that we can start working on right now to lower our odds of developing dementia. I would like to acknowledge the help of my team, Terry, the Brain Health ARC, GAP for providing the data, all my Bio-Hermes collaborators from across the UK and also Leiden in the Netherlands, and of course all persons affected by dementia who make this research possible. Thank you.
Okay, quick fire's done. Phew, it was quick. We now have our keynote speaker. I'm very pleased to welcome Will Whiteley up onto the podium. So Professor Whiteley is Professor of Neurology at University of Edinburgh, clinically works very much in the stroke space, but has been invited here because of the really amazing work he's been doing around collating data, particularly NHS data, and making that available for research. So Will, over to you. Thank you.
Thanks a lot. Can everyone hear me? Just double check which thing I'm using. Well, thanks a lot for the invitation. So as Terry said, I'm a neurologist, but I've been working recently with Health Data Research UK to try and think about how we can use UK-wide and Scottish, particularly, data for a number of questions. And my talk, I'm going to talk to you about, I'm going to start off by giving you a general overview of where we've been working across cardiovascular disease, and think then a little more about how we can work in dementia and neurodegeneration. So here are my funders, and they're all pretty much academic, other than a small bit from Eastside. So what might the use of whole population data from the NHS do for us as researchers? Well, whole population cohorts might help us understand disease experience without selection bias. So clearly, UK Biobank and Our Future Health will be very, very important resources for all of us as researchers, but there's the consent bias of going in. And because all of us in the country use the NHS, that disease experience is pretty much without bias. We could link some of those mega-scale cohort studies to electronic health records to give us a better idea of disease outcomes. UK Biobank and Our Future Health do that already, but we can do more to better phenotype the outcomes they have. We might be able to recruit to clinical trials and measure outcomes in those trials at lower cost, and there are certainly examples of that. And of course, we've all heard about AI. Perhaps we could use these mega-scale resources to develop really stronger AI in imaging or in records, to understand diagnosis or make diagnostic algorithms, think about prognosis, or do things that might improve health systems. But to do this, what we need really is collaboration. These don't work as as research done in institutions individually, collaboration across institutions and collaboration across disciplines is very, very important. Because we're using data that's provided through the course of healthcare, where people can't avoid leaving a data trail, we need to make sure that we're engaged with patients and citizens about use of data throughout the process. And because much of our work is going to be publicly fundable, we need to make sure it's available for others by making the data analysis findable, accessible, interoperable, reusable, or fair. And what the move is those of us who've worked with perhaps an older generation of researchers who always thought of data as research power, my data is mine, you can come and work with me, more to thinking about data as a community resource, much in the way that BioHermes has worked for us today. So I'm going to give you the example of a collaboration in cardiovascular. So the collaboration in cardiovascular is the British Heart Foundation Data Science Centre, and that aims to really look at 6 themes to try and bring forward research in these areas. And the themes are whole population data, where imaging data, where the Scottish Medical Imaging Dataset is the jewel in the crown, wearables, defining disease, so how do we see what that trail of breadcrumbs through the electronic health record looks like for people with different diseases, so we can do better to understand people's experience, linking cohort studies, and lastly, perhaps the holy grail of doing data-driven, data-dependent, electronic-only trials. So these are the questions the Data Science Centre is trying to work on, but there are also a number of, which are methods, but there are 3 disease-related cohorts, one in diabetes, kidney, stroke, and more recently one in motor neuron disease and respiratory. So the British Heart Foundation sounded like a great idea when it was set up in 2019, but there wasn't really anything to give it a push. And then in November of 2019 and January of 2020, a COVID-19 pandemic came along, and here were the first reports, which really were cardiovascular complications of the COVID-19 pandemic. And suddenly, large-scale population-wide linkage of electronic health records to cardiovascular outcomes became of nationwide and even international importance. And the UK was one of the few places where this was possible to do at scale. So here are a couple of examples of this work. So this is now creating cohorts of people, of everybody who's registered with a general practitioner at the beginning of 2020, and looking to see if they have an infection of COVID, defined either in the dark red as hospitalized with COVID, and in the light red as simply having a positive test, looking to see their risk of stroke and myocardial infarction on your left, and pulmonary emboli, deep vein thrombosis on your right. And these are hazard ratios. Each point is a hazard ratio telling you how many times more likely you are to have a heart attack, or stroke, or DVT if you're infected rather than not. And what you can see is that the risk of heart attack and stroke, arterial thromboses, is high early and declines rapidly, and that's much higher in people who are hospitalized versus not hospitalized with COVID-19. And by the time you reached about a year, that had reached the population level incidence. But if you looked at pulmonary emboli and deep vein thrombosis, venous clots, that was rather different. There was a much higher risk in people who are hospitalized, but the risk seems to maintain up to about a year. An unexplained risk, but you also see this in previous influenza pandemics. And so there may be something causal here that we're missing that's worth researching. So that's an example of what could be done, and that was done only in England. But of course, the whole population of the UK is about 60 million people, and the vision has always been, can you do studies across the whole population so we can understand the entire variety of people's disease experience by region, ethnicity, deprivation, and so tell us something more than we have in any one country. So this analysis, led by Aziz Sheikh in Edinburgh, now Oxford, and Kathy Sudlow of HJI UK, did the same analysis looking at the risk of not being vaccinated with COVID-19 by different people's characteristics. So each of these little tables is each of these points in the tables represents as an odds ratio of the likelihood of not being vaccinated. So, for example, if we look here, and each of the things is by age group with increasing age, and the top few figures are for ethnicity, and the bottom few figures are for deprivation. So what we can see if we look by ethnicity, people who have a black ethnicity were less likely to be fully vaccinated than people with other ethnicities, and that was similar across regions. And deprivation was a strong predictor of vaccination, where people who were least deprived were less likely to be vaccinated of all the other deprivation groups. And so what that did was this directly informed ongoing vaccination policy and informed us about who were the important unvaccinated groups across all the countries of the United Kingdom, and really was the first of those federated analyses. So this is the kind of thing that's possible, and something we should look forward to when we're thinking about other public health challenges. And of course, going forwards, a major public health challenge for all of us is dementia. So why was this possible? Why was it possible to do these linkages during COVID-19? So our if you like, our pillars of being able to do this are that you have a governance structure, that you have data that's available, you have data that's curated, you have a collaboration of researchers, you have citizen support, and there's innovative data access. So during COVID-19, all these things faced in the right direction. Governance was rapid. There was no 6 weeks to wait for an ethics application. There was no 2 months to wait for an approval by PBPP. Data became available across system, and for these purposes, data became available from primary care. Data was curated in a number of research groups, so Open Safely, a Thin research group, and that's the QRisk research group, and the BHF Data Science Centre all put significant resource into curating research so that research could happen more, curating data sets so things could happen more quickly. There was widespread collaboration. We all remember that's the time where Teams and Zoom really took off as a way of working across different sites. There was widespread citizen support, and there was innovation in data access. So the NHS England developed its own trusted research environment, like the secure data environment in Scotland, which had been present for some time. And Open Safely, led by Ben Goldacre, had a new mechanism where access to data was only by containers, where researchers could never see primary care data, a model we think is going to be what NHS England, at least, does in the future. However, after COVID-19, things have changed. Government's gone back to its old pace, which means it's slow, and that's largely a resource problem rather than lack of willingness. We're losing access to some data. Certainly national primary care data, at least in England, won't be available. Currently is not available in Scotland, but might be by 2016. It continues to be available in Wales. Data curation is supported. BHF Data Science Centre continues to do this. I think there is now a widespread change in research culture of collaboration across institutions. That's much easier, and I think business as usual. Citizen support, I think, needs to be maintained. So during this time, citizen support was taken as read because we were dealing with a health emergency. But from now on, we need to continue to engage at all levels and find people who disagree with us. There will be some people who will never want any piece of data access. There are some people who want access without any restrictions, and so many people in the middle who we can think about sharing benefits. And these innovations in Open Safely and NHS England are continuing. And I think Open Safely will be particularly the way that many of us, and maybe in models in Scotland too, the way that many of us access primary care data. Right, so that's a bit about what the cardiovascular research, why COVID was perhaps an impetus to getting this kind of thing done, and how we access data. What does it look like across the country? So in each of the UK's 5 nations, there is a secure data environment. In Scotland, it's the National Data Safe Haven, run by Public Health Scotland. In England, it's the National Secure Data Environment, run by the NHS England, no longer exists, of course. Quite what's going to happen to this, we don't know. Accessible through a DARS process. Open Safely is run from a collaboration called the Bennett Institute in Oxford. In Wales, there's a long-standing collaboration called SAIL, and that continues. And in Northern Ireland, the Honest Broker Service, which is just setting up and relies on, is going to rely on some legal changes which will depend on the Northern Ireland legislative body, which needs to do more work. And then across the whole of the piece, the Office for National Statistics also holds a secure research data service, which is less detailed. So there are all of these things where we can access data, as well as in England and in Scotland, regional safe havens. So us in Scotland, there are 5 secure data environments, which personally I feel is crazy, but we can go to each of those. So we know where we can do it, and those are now reasonably accessible and have a reasonable compute resource. But if we're going to do things and understand people's disease experience, we really need to make sure we're linking across health. So here are some of the data sets where it's possible to link now, and I think more of these will become business as usual in future. So data from hospitals and death records have been available for some time, allowing us to do those kind of studies. Those are relatively low resolution, but do tell us something about people's dementia experience, for example. Community pharmacy and other data has been available since 2016, so we know when people either are prescribed or pick up their medicines. We have data on screening, and linking that to these data resources will be important routinely. It's not currently available routinely, for example, for bowel cancer screening. Lab data is available in Scotland, although more work needs to be done to link blood test data routinely to other data sets, although it is possible to do it, and people have had exemplars of that, some of whom are in the room. Then audits and registers, so stroke registers, which has got very highly phenotyped information, myocardial infarction registers, vascular surgery registers, all can be linked as well, telling us a little more about disease phenotyping. For us, who are interested in neurodegenerative disease, dementia, and the related disorders, and Parkinson's and motor neuron disease, social care is really important because that's the major cost of these illnesses, and this gives us some level of disability as well. These are available in smaller regions of the UK. Some regions are digitized, other regions not. So currently the research on social care is only available in particular places because council services aren't as digitized as health. But it's only by linking these things together that we're going to be able to understand people's whole disease experience. And I think the Sudlow review published just at the end of last year will make much of this, again, possible and easier. Why is linkage important, say, for disease ascertainment? Well, linkage is important for disease ascertainment because often disease only is recorded in one or other of these data sets. Because if we think about recording, say, here we've got 6 illnesses: depression, anxiety, COPD, osteoarthritis, epilepsy, and visual impairment. The recording of these is sometimes by chance. So here, this is data from the UK Biobank, where the ascertainment is in different circles. So GP records, as you can see, in every single condition, is the largest single source of disease information. There's much less information in hospital records. And if you look in UK Biobank, some people are doing self-report, but many fewer. So primary care is, if we're interested in disease ascertainment, particularly for illnesses that are largely looked after as outpatients, like the neurodegenerative diseases, we do need to think about how we access primary care. So that's telling us about linkage systems, patients and public. So really, without public trust, using health systems data is impossible, really. So you might remember back a few years ago where there was the attempt to bring data together under something called care.data, and because there was insufficient public engagement, and there are insufficient plans for the relevant objectives. That entirely failed. But now we need to work really quite slowly to make sure we're building trusted environments. Open Safely is one that particularly seems to be accepted by computer data security experts, but make sure those are available for people to use. And it's not a one-off to engage with the public, it's continuing talking about new developments, results, public benefit. So frequent and real engagement about data uses, about benefits, and about individual research questions is super important. That doesn't have to be days and days, as long as you're informing somebody and getting different people's views. And then data controllers and researchers need to engage with those responses. So often they will shape your research, shape your research. Many members of the public are really positive about use of data. A few, I think, in my experience, a minority, but very much an educated minority, are not in favour. And we need to engage with both sides because there are difficult questions. Engagements with industry, how much, where, to whom, and how much financial benefit should come from the research done in these data. What about sensitive topics, mental health problems that people don't want to share, such as schizophrenia? But there isn't a final answer to this question because this is a political and engagement part of what we do. Okay, so I've told about COVID-19. Let's think now what we're doing with these currently. So I'll tell you about estimating some chronic disease epidemiology, then thinking about some brain examples in neurodegeneration and stroke, in randomized trials and hospital systems. So, first of all, what can we do when we're looking at an entire health system's the patient experience across an entire health system? What new things can we look at? Well, this graph here, this shows you all of the, well, pretty much anyway, 79 cardiovascular diagnoses as defined by ICD-10, and gone through by me and my colleagues to try and define. And we're seeing how, and each square represents a relative risk of this condition by each of these 17 ethnicities, all relative to the white ethnicity. So we can look at experience in much finer grained detail than previously really has been possible looking at national-wide data. Usually people looked at larger, data sets have been smaller, so people have looked at much larger ethnicity categories, but it's very clear that experience of disease varies by much finer ethnicity categories. So if we look, the red points are where people are having more disease relative to the white population. The blue points are where people are having less disease relative to the white population. We chose the white population because it's the largest. So you can see in this group, which are usually defined as Asian, people of a Chinese ethnicity have a much lower incidence of all cardiovascular diseases compared to white population. But if we look across the people defined as South Asian, it's clear that people who are of Pakistani ethnicity or of Bangladeshi ethnicity have the highest risk. Taking people together and realizing their community experience and now taking this into communities. Of course, this work is being done already, but this helps to highlight it, is important. And then within people who have a black ethnicity, we can see there are two kind of hotspots of red in large artery coronary disease and in hypertension. And then for hotspots of blue, really in venous diseases, which seem to be common in people of a white ethnicity. So we're going to work towards doing that across now deprivation and region and start to think how to do that across the range of neurodegenerative diseases. And if we look by region, so we can look here, this is the Integrated Health Board or Health Board equivalent in England, and we can see how different diseases have different regional patterns. These aren't necessarily new, but what is new is the extent of adjustment we're able to do for relevant confounders like deprivation, ethnicity, and other things. So peripheral vascular disease, particularly deprivation associated, much commoner in northern England. Ischemic stroke, particularly age associated, particularly commoner in coastal regions of England. So that's thinking about how we can do things generally. Let's now think about how we can measure quality of care. So this again is relevant to dementia because national measurements of quality of care, Terry's making great efforts to do that for dementia, but having this routinely and easily available is very hard. So here what we're trying to do is to add new measures of quality of care that aren't currently accessible in current registers or audits. So everyone who has a stroke who comes to hospital, details are filled in about them, usually by clinicians, which is very time intensive, and sometimes time intensive takes a lot of time, and there's actually still a lot of manual inputting of data. And that only tells us about people's inpatient experience. It doesn't allow us to follow people very well into the community. So here we're looking at a current public health priority, which is the prescription of secondary preventative medicines to reduce the risk of subsequent stroke or myocardial infarction, which we know is poor across the piece of almost all cardiovascular conditions. And we had no real way of looking at this for people with stroke in a year after their illness. So just to show you what we've got here, and we've got 5 plots. Each plot represents a different medicine or group of medicines. So lipid-lowering drugs, which are essentially statins, antihypertensives, blood pressure-lowering medicines, antiplatelets, aspirin, clopidogrel, and the like, anticoagulants, largely given for atrial fibrillation, and a combination of these 2 blood thinners, anticoagulant or antiplatelets. So what we can see is that if we're looking at someone giving some kind of blood thinner, and this is by category, so age, sex, and ethnicity. So if we think about, the first thing you can see is that there isn't that much difference by people's categories, just looking generally across these graphs, apart from age. Age is the big one, but fortunately, prescriptions of secondary prevention doesn't seem to vary too much by age or ethnicity, which is good, because it shows us we're not discriminating, but it does tell us that if you look at the overall proportion of people who are prescribed, for example, antihypertensives, over the whole piece, prescriptions of these medicines is not as good as it should be. This should be up at least at 80%, and it is currently at 40%. Lipid-lowering drugs, which should be there for pretty much everybody with ischemic stroke, is down at the 75%. So we're doing poorly across the piece, and that allows us then to to advocate for change in practice. Okay, so we've thought about a whole population, some kind of ways of looking across the population. We've thought about measuring quality of care. What about the very long-term follow-up of clinical trials, where we can use that really valuable thing in a clinical trial, which is a randomly allocated medicine, which allows us to draw causal inference. So here's an interesting question. What is the effect of the drug metformin on the later risk of developing dementia? Yeah. So Sam Altman of ChatGPT and OpenAI, he takes metformin every day and hope that's going to stop him from getting dementia. Yep. Is he doing the right thing? So the problem is, in clinical trials, by and large, people who are in clinical trials usually are people in their younger years, and by and large, if you take people without dementia, too few people to develop dementia to detect an effect on treatment. So the treatment, the duration of the trial is at most 5 years. In this particular study we're going to talk about 10 years, at most 5 years, and not enough people develop dementia during the trial. Maybe if we followed them up to their later years, we could see whether this short period in midlife, where we think that most treatments should have their major effect, would be detectable. So can we extend this follow-up? So I did this with, my late mentor, Robert Turner, set up this trial, a UK prospective diabetes study, started in 1977, and he set it up to find out what the effect was metformin on the risk of developing diabetic complications. Can we repurpose that by linking people in that original trial to their electronic health records to see if they develop dementia? So this was done with colleagues in Oxford, in the Nuffield Department of Population Health and the Diabetes Trials Unit. And so the trial reported at 10 years the effect of metformin on death, on mortality, and on dementia. Dementia wasn't recorded during the study. And so what we did by linkage was, this is currently very early results. We've just got additional data to come in from Northern Ireland, We then linked people to see the incidence of dementia over time. On the right over mortality, we can see that for people with diabetes anyway, the prescription of metformin reduced the incidence of death by about a quarter, really leading to an increase in life expectancy of a couple of years. So maybe Sam Altman is doing the right thing. And there was a reduction in dementia incidence of a similar magnitude, but too few cases for us to determine that with any certainty. We're now searching for new cases just to add to this analysis. So we can do that. We can look at treatment midlife, look to late life. What about understanding free text and images, which we know have got really important information for neurodegeneration? So we can do this in Scotland. So in Scotland, Scottish Medical Imaging copied all of the data, all of the brain imaging, all of the imaging data was taken through routine clinical practice at the moment between 2008 and 2018, soon to be up till 2024. And took it into the Scottish National Safe Haven, where it's possible to link to neurological data from registers and from hospitalizations, deaths, prescribing, which I've told you about before. And we've got a secure place where we can do anonymized linked imaging analysis. We've got some bespoke ways of for preparing that data, prepared it for in terms of identification of sequence and preparing those images for AI analysis, and secured analysis and a mechanism for exporting models and exporting tabular data. And compute at the moment, which is 1 GPU per virtual machine, but soon to be 48 GPUs. So what can we do with this? Well, first of all, we could do natural language processing. So you'll be familiar with natural language processing. Now you'll see your pop-up on your Google search, which comes from a large language model, which is a type of natural language processing, ChatGPT similar. We're now using, we were using, when we started this a few years ago, a much simpler rules-based natural language processing, which includes negation and lemmatization and other techniques, which allows us to take, if we're interested in the condition of cerebral small vessel disease, which is the substrate for vascular dementia, as well as increasing risk of Alzheimer's disease, we'd like to identify this at scale. And so what we'd need to do is first of all, to try and get some of this information from brain imaging reports. So here's a report that a radiologist has written about a patient, and there are at least 650,000, possibly 1.5 million of these. And so however eager your medical students are, you're going to have an army of them if they're going to replace natural language processing, which is the way I started this. And unfortunately, I all got very bored and pissed off with me. So that's why I turned to the computer. So what we've got here is the results of this natural language algorithm, which identifies this brain image report has small vessel disease, has an old infarction in the cerebellum, a deep infarction, and another sign of small vessel disease, a lacume. And this then turned this report into tabular data that's suitable for analysis. So we've got about 650,000 people. There's about 1 million scans, and that gives us a cohort of about 225,000 people with cerebral small vessel disease, a couple hundred with cerebral atrophy, about 150,000 with ischemic stroke, and about 9,000 with a rare disorder, benign brain tumor, meningioma. And we can start to think how we can analyze those. So the first question, the reason I started this, was to say, well, how does asymptomatic cerebrovascular disease, so-called covert cerebrovascular disease, influence later dementia and stroke? There are a number of bespoke cohort studies examining this, but none of them are really large enough to look at interactions. So does this vary by age? And none of them really apply to real-world reports that those of us who are clinicians get in our inboxes every day. And so on your right here, we've got a forest plot which tells us in bold each outcome: stroke, dementia, and colorectal cancer as a control condition. And here we can see that there is a differing pattern of association of risk for different vascular markers on brain scans in people who've never had a stroke for both stroke and dementia. So stroke, for example, people with old and never symptomatic cortical infarcts have a higher incidence of stroke than people with the other characteristics, whereas if we're looking at dementia, it's atrophy and small vessel disease that are the clear things that increase risk. Small vessel disease, not shown in this diagram, particularly increases the risk of dementia recorded as vascular. We were worried about bias, and that's why we've included the control condition of colorectal cancer, where there is no association, also a disease-associated, an age-associated condition, like age and dementia. The second thing is then, well, we've identified people with this condition, but asking people with the relevant biomarker for vascular dementia who don't have symptoms, covert cerebrovascular disease, 2 trials is difficult to do at scale. And so we've now started an exercise funded by the Chief Scientist's Office to go through some of that ethical and public engagement piece to try and see how acceptable it is for us to write to people to tell them they've got covert cerebrovascular disease, or would they like to take part in the first instance in a cohort study, moving then to trials. Right, so we've looked at reports. Reports are fine. But what we really want to do is look at the images themselves, so we know that's much more detailed. So there is currently a project funded by these 5 organisations and a collaboration between these 2 universities aiming to predict the incidence of dementia based on 3 machine learning models, comparing their performance, but using image-based machine learning models to predict later dementia incidence. Whilst we aren't going to be producing models that are really ready for prime time for clinical practice, what we are doing for you lot is cleaning the pipes so that future AI analysis is possible to look at all sorts of different questions. So we've developed a dataset called the Brain Health Data Pilot, which is open for applications. So this is an evergreen dataset with streamlined access for people who are interested in pre-competitive research with a streamlined application. So it's only about 4 pages, where user-generated outputs are stored. So if you're not experienced as an imaging-based researcher, imaging-based phenotypes are available. For example, brain volume will be accessible for international users. We're progressing that. GPU cluster for analytics expected probably by June, and continuing patient involvement. And so that's useful for 2 things. First of all is to enrich these for epidemiological analysis. So we've got the NLP phenotypes ready just now. Pixel-based phenotypes are in progress, and linkage to cohorts. And then we'll try and reduce time to AI, because AI researchers and image-based researchers are 2 groups of people. So we will be preparing images according to the best standards of imaging-based researchers, then to be used by deep learnings and the rest. How can we work and improve our machine learning across Europe? So I won't go into this too much, but we have a collaboration with Helsinki, where there's a similar sized population to Scotland with a similar approach to data regulation, where we have a data visiting principle for federated learning, where we'll try and do our dementia prediction experiments in 2 places, and we'll have an AI model that moves between places to improve and learn from both. And if that works, then we'll be going to Bonn and Bordeaux for further work. Okay, so that's talking about AI and how that might be useful for us as imaging-based dementia researchers. Lastly, let's just think quickly about how we can use data in general to make trials quicker. So this is the Dementia Trials Accelerator has been funded by MRC through the Office for Life Sciences, and aims to enhance some of the functions of joint dementia research in the Scottish Dementia Registry by enhancing the amount of biomarker information that's available to new trialists, and that biomarker information, of course, is one of the key things that new industry trialists want to see. So we're developing single-shot window. We're developing ways that people can have biomarkers measured in a similar way to those, many of us had vaccinations during the pandemic, and we will be trying to deliver this hybridly using the collaborations of the NAHR and the other dementia delivery networks across the United Kingdom, And we're starting to use people from cohorts, to invite people from cohorts in the second half of this year. So what we'll aim to do is have NHS and population cohort partners, try and find a targeted population for contact, so an age range and disease range, and then add community biomarker and cognitive screening to that invitation, so that if someone wants to take part in a trial for industry or a trial from academia, that can happen much more quickly. So there's a lot we can do with nationally available data, and this is an area that is highly active. Health and data science certainly aren't the answer to all these problems, but it is a new lens to look at these problems. And why not have a new lens to try and understand things we don't know already? But if we're going to succeed, us as health data scientists, we do need to be led by citizens, because we are using data collected as part of clinical practice. We do need to be led by the science as well, to make sure that our analysis are relevant. Training the next generation, there are many resources for that. We need to be collaborative and open rather than closed and private. And if we're going to work, there are things that need to happen in regulation and compute resources that are really important over the next few years, but I think those will come. Thanks very much.